Hello everyone, hope you all are doing well. So today we are going to discuss the next answer copy of our Aim for Agriculture series. So as you all know that we are having Aim for Agriculture series in which we are having daily answer writing practice and weekly full tests. So today we are going to discuss the copy of day 41. We have already discussed the copies till day 40 and today we will be discussing the copy of day 41. And by analyzing this type of copies, you can also get to know how to approach your answer, how to introduce, how to conclude, how to write certain points, how to use certain examples, how to structure your answers. So overall these answer copy can be very helpful to you in your answer writing journey also. So let's start. So the first question is, discuss the role of DNA fingerprinting in plant variety protection and identification of cultivars. So we all know what DNA fingerprinting is. It as we have as we all have different fingerprints. So DNA fingerprint is also different in all the species. So through DNA fingerprinting, we can identify various types of varieties. We can protect various types of varieties because it is a unique genetic pattern of different plant species and cultivars. So we all know what DNA fingerprinting is. Now, how is it helpful in plant variety production and identification of cultivars? First of all, uniqueness verification. Okay, we all know that every DNA is unique, so through that you can verify. It can help in legal protection. Many times the breeders can be using these fingerprints as their signatures. Okay, so this can be done. Can be helpful in enforcement of breeders right. So as I will be discussing every point in brief, मैं जो भी points है सबको brief में discuss करूँगा. So if you want to read the answers, you can simply pause the video and read the answers in detail. Okay, if you want to. So next point was enforcement of breeders right. Obviously it could be helpful in enforcing breeders right. In identification, obviously it can be helpful in cultivar differentiation. Seed, it can be helpful in seed purity verification. It can be helpful in tracing cultivars origin. It can be helpful in analyzing the ancestral relationships with the ancestors of cultivars. So in all these things, the DNA fingerprinting can be very helpful. So very well written answer, limited points but well explained. So I hope you have read the answer and you have got certain idea. I think examples are also well mentioned. You can also read these examples and you can also add these examples in your notes if you haven't add examples in that. Okay, so through this you can also value add it to your notes and again appropriately concluded. Now let's move to the next question. The next question is, what is Technology Assessment and Refinement Program, that is TARP? Discuss the methods of assessment and refinement of new technology. So as the name suggests, Technology Assessment and Refinement Program. So it is an initiative to enhance the technologies present in agriculture. Okay, as the name only suggests, we have to refine the technology, we have to assess the technology and refine it. Now, how can we assess the new technology? More methods: by doing on-farm trials, okay, by doing participatory rural appraisal, through which farmers can participate in this technology assessment method. We can do socio-economic impact assessment, how the technology is impacting the farmers. Are they getting real benefit or the benefits are only on the papers? So those things can be very helpful. How can we refine? Obviously similar points would be there, that is adaptive research trials. We will do various trials, we will do various field trials. We will see the farmers how they are adopting to it, is there is any problem in for farmers in analyzing and maintaining that technology. Everything needs to be evaluated. Farmers feedback is very important. Pilot projects and scaling up, we have to take several pilot projects to see is the thing is really working. So all these things are needed to be done. And again, fairly written answer. I hope you are reading the answers at several points. Now let's move to the next question. The next question is, what is jelly? discuss the principles and problems of jelly making, and briefly describe the method of preparation of guava jelly. So many times what happens, many students skip these types of topics, that these topics are not that important, but these questions have been asked in UPSC previously. अगर आप previous year papers देखो तो you will find these types of questions. Okay, so that is why you have to give proper amount of attention to these types of questions also. So what is jelly? We all know that it is a firm, means jelly, you all know that not liquid and not also solid. So a thing between the liquid and solid mode is kind of jelly. I hope you all have seen different types of jellies available in the market. You have eaten different kind of jellies, and these jellies generally prepared from the pectin, strained juice of pectin, and they are set through by cooking. And for example, guava jelly, mango jelly, tomato jelly, different kinds of jellies are present in the market. Now, what are the principles of jelly making? First of all, fruit juice selection. We have to select proper fruit with a high amount of pectin content, because this pectin is very important for jelly making. So pectin content should be at appropriate. Acid content should be right at right levels. Sugar content should be at right levels, light levels. Okay. Cooking process. We have to understand the cooking process. So these are the some of the principles in jelly making. You can pause the video and read these points. Now, what are the problems in jelly making? There are various problems also. Many times, what happens when the pectin amount is not up to the mark, the jelly doesn't set properly. So sometimes you will see the jelly is failed to set due to insufficient pectin or acid. Okay, cloudy jelly, the jelly will be not in perfect shape. It will be a cloudy type of shape, which is very hard to strain. Okay, weeping is another problem, that is presence of excess liquid. Crystal formations, many times what happens due to use of excessive sugar, those sugar crystals can be formed on jelly. So these are some of the problems which we encounter during making a jelly. Now, how can we prepare guava jelly? First of all, we will select proper guavas, proper ripe guavas with appropriate pectin content. Then we will do juice extraction. Then we will cook guavas with sugars so that the sugar and acids are in appropriate amount. We will then taste it and then let it set, and we will see that it sets properly. Okay, there are no crystal formations, there are no excess liquid. So all these things needs to be seen. So, well written answer. The brief points are discussed in brief, and everything has been well mentioned. So I think you like the answer. You have read the answer by pausing the video at several intervals, and these types of answer copies could be very helpful to you in your answer writing journey also. So that is all for today. If you like it, please press the like button. Please do share, and please do subscribe. It takes a lot of hard work to make such videos for you on regular basis, to bring such courses for you at so much less price. So it takes hard work, and if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.